my name's Sean. I'm from SMS, and welcome to this webinar. I'm introducing Peter Budd, who's here today to give a speech. Peter did his bachelor's and PhD at Manchester, and then did a brief stint at the British Petroleum Research Company, and then went on back to Manchester to do a lectureship and professorship in polymer chemistry. And his research has been very expansive in working with polymers of intrinsic microporosity in areas such as energy storage and carbon capture. So he's going to give a presentation about those materials today, and I'm looking forward to it.
Thank you very much indeed. So it's a real pleasure to be here and to be in London on a day when it's actually snowing outside, which I hadn't expected. And I'm Peter Budd, and I'm from the Department of Chemistry at the University of Manchester. And I'm going to be talking today about the polymers of intrinsic microporosity, or PIMs, and in particular about their membrane applications. Now, we're thinking about separations, and separations on an industrial scale. And a few years ago, David Scholl and Ryan Lively wrote an opinion piece in Nature where they pointed out that a lot of chemists and chemical engineers spend a lot of their time actually trying to separate mixtures. And of course, if you look at a refinery or a typical chemical plant, you see lots of big columns, and what they're doing is separations by and large. And often those separations are based around distillation, which is a very energy-hungry process. Indeed, it's been suggested that 10 to 15% of the world's energy consumption goes into the separation of chemical mixtures. And what David and Ryan suggested was that there needs to be more of an emphasis on newer technologies, which can be much more energy efficient. And membrane science represents one of those areas where there is the potential to have much more energy efficient separations and new types of separations if we can get the membranes that do the job well enough for us. So what I'm going to do today is, first of all, because not all of the audience knows about membrane science, give a little introduction to membranes and membrane processes, and what we're looking for if we're trying to develop a new material for membranes. And then I'll move on to my favorite polymers, the polymers of intrinsic microporosity, or PIMs. And after a bit of background, I'll focus on three areas where there's a lot of current research, where we're thinking about the topology of the polymer and how that may affect the properties, where we're modifying the polymer to tailor it for particular separations, and also where we're combining the polymer with other materials in what are often called mixed matrix membranes. So there's an outline of what we're going to do today. But if we start with membranes, so we're thinking about, in the simplest case, a thin film of something which acts as a selective barrier to particular molecules or particles. So if we have a feed mixture, which is a mixture of different components, the stuff that goes through the membrane, that permeates, will have a different composition to the stuff that doesn't. And these membranes, they can be in the form of flat sheets, they can be hollow fibers. Of course, one type of membrane many people will be aware of is the biological membrane that surrounds every cell in our bodies. But here I'm focusing on membranes that can be used on an industrial scale, potentially to replace some of those very energy-hungry separations that we mentioned earlier. And in the simplest case, in the research lab, often what we're dealing with, because we want to understand the material, is a relatively thick, and when I say thick, I mean sort of 40 to 100 micrometers, which in these terms is thick, a relatively thick film of something. And I'm a polymer chemist, and so we're often dealing with polymers. That is to say, molecules made up of a large, smaller units. But of course, other materials can also be used. We have ceramics and other materials that are formed into membranes. But if you're an engineer, you want as much stuff to go through your membrane as possible, so you can have the minimum membrane area. And usually that means you want a very, very thin film, often less than a micrometer in thickness. And if you've got a very, very thin film, it's not going to be robust enough for use without a support. So a real industrial membrane is generally much more complex than a simple film of a polymer. it has a structure where we have a very thin separation layer and then a support which gives it robustness in use. And there are different ways in which that can be achieved. In some cases, if it's a polymer membrane, we're talking about an integrally skinned asymmetric membrane where we take a polymer in solution and we precipitate it in a way that we have a very thin film which is relatively dense and performs the separation on the outside, and you end up with much bigger holes on the inside which provides support but doesn't have too much resistance to transport. And this is made as hollow fibers or as flat sheets, and many of the current membranes for gas separations are actually integrally skinned asymmetric membranes. But sometimes it's not very easy to do that with an expensive polymer and process it in that way. So another way in which you can get a similar effect is to have a very thin film which has either been coated or in some cases polymerized in situ on a porous support, which is another material. So this is then a thin film composite or TFC membrane. And I'll be talking more about these types of membranes in the talk. Now, there's a whole range of processes that we can use membranes for. And if anyone who's done chemistry at school has probably used some sort of membrane process, because you use a filter paper to remove particulates. But in our research, we're interested not just in taking particles out of solution, but separating molecules. We're interested in separating a molecular scale. And so the sorts of processes we're interested in are things like nanofiltration, reverse osmosis, pervaporation, and gas separation. I'll say a little bit about some of these, but in particular, pervaporation and gas separation. But first, let's just mention reverse osmosis, because this is an important commercial application of membranes. One of the global challenges is how to get clean drinking water for the global population. And in many parts of the world where people are close to the sea, one way to get drinking water is through desalination. And currently, most new desalination plants are membrane processes. They're based on reverse osmosis. And in reverse osmosis, your membrane is a semi-permeable membrane which will let the solvent, if it's water through, but prevent other things like ions from going through. And in reverse osmosis, you need a driving force. In any membrane process, you need a driving force. In reverse osmosis, the driving force is obtained by applying a pressure on the feed side, which is greater than the osmotic pressure, which is actually pushing things in the opposite direction to what you want. And so that's a current process which is increasingly used for desalination in many parts of the world. But I'm not going to talk any more about that today, but I will mention a couple of other processes. One of them is pervaporation. And pervaporation, the word is a combination of permeation and evaporation. Here we have a membrane. Again, it's a liquid mixture, which is our feed, but we remove the permeate as a vapor. And we do that, we create a driving force for that, either by applying a vacuum on the permeate side or having an inert sweet gas, which removes the permeate as a vapor. So there's a phase change on going through the membrane from liquid to vapor. And I'll say a little bit about that more later. And then another one that we've been very interested in is membrane gas separations, where feed is a mixture of gases, and both the permeate and the retentate are in the vapor phase, but we have a difference depending on different permeabilities to a membrane. And again, we need a driving force, which is a pressure gradient. We can achieve that either by pressurizing the feed, or in some cases, it's better to reduce the pressure on the permeate side. So those are membrane processes, some of the membrane processes that we're interested in. But if we want to create new materials so that these processes can be applied either more efficiently or in ways we can't use them at the moment, there are a number of things that we need to deal with and to think about. And of course, any membrane material, going to use it to separate things, it must be able to separate. There has to be selectivity. There has to be a preference for one thing to pass through the membrane rather than the other. But also we need to get a lot of stuff through the membrane. Otherwise, it's not going to be a commercially viable process. And so we have to think about those two things. So we can have selectivity and something which I've described to as permeability. Now, there are a number of different words, we'll meet another one later, to describe how much stuff gets through a membrane. In the simplest case, we can think in terms of the flux, which is the amount per unit area per unit time. But that, of course, will depend on the driving force. And so when we want to compare membranes, we often actually normalize that with respect to the driving force. If it's a gas separation, that's an effective pressure difference, to get something that we'll call permeance. But that in turn will depend, if it's a simple film, on the thickness of that film. If it gets thicker, less stuff goes through. And so we can also try to normalize that for thickness to come up with an expression for permeability. And when we think about a material, we often discuss it in terms of permeability. But when we think about a membrane, particularly one of these complex membranes like a thin film composite, we'll discuss it in terms of permeance. So we'll see both of those things come up later in the talk. But the thing about selectivity and permeability, and one of the frustrating things if you're a researcher trying to develop a new material, is you can put a lot of effort in trying to improve selectivity, and the trouble is you find you've lost permeability. Or you see, like, you want something to be really permeable, and you find you've lost selectivity. So there's a balance, a trade-off between permeability and selectivity. And that's one of the first things we have to tackle. And usually in practice, we're finding an optimum balance, because we need a certain degree of selectivity. In fact, these days we're trying to push up the selectivity, because one thing we have done in our research is to find ways of making membranes that are much, much more permeable than we used to think was the case. But as well as selectivity and permeability, there are lots of other things we need to think about. We have to be able to make our membrane, fabricate it on a large scale economically. So we need processability. We've already indicated that, if you just have a very thin film or something, it's probably not going to survive the conditions of use. We need good mechanical properties. And that is why we're often dealing with composite materials or things which have complex structures in order to give us the combination of permeation properties and mechanical properties that we need. We may be using this membrane with all kinds of contaminants present at times at high temperature, and the membrane needs to survive the conditions of use. We have to think about chemical and thermal stability. And then there are all sorts of things that can happen to a membrane in use. And we'll see a bit more later when we think about gas separations and we're thinking about certain sorts of polymers. There's a process of physical aging which occurs inevitably that changes the properties over time. If we think about liquid separations, in particular water treatment, then fouling is always a big issue, whether it's biofouling, the building up of algae and other things, or the build-up of inorganic materials, just like particularly in the London area, your kettle scales up when you use the water. And so aging and fouling are things, one way or another, that one has to think about. So we'll say a bit more about aging and other things later. So I'm a polymer chemist, and so I'm particularly interested in polymers that can be used in membranes for a variety of membrane processes. And often the polymers that we're dealing with are glassy polymers. So a glass is something which has an amorphous structure. There's no crystallinity, no regular arrangement of molecules. But and normal polymers will form a glass at low temperature. If you drop the temperature, you get to a point where there simply isn't enough space in the material for the big polymer molecules to be able to move around. They're locked, more or less, into a particular situation. So it's not a crystalline structure, there's no order there, it's amorphous, but it's glassy. And of course, the thing that we call window glass is an inorganic example of such a material. But the thing about a glass is that it's not an equilibrium structure. It's the process of forming a glass is dominated by kinetics, by dynamics. And so we've sort of locked what is really a liquid structure into a sort of solid-like state. And what we're trying to do here is to use the bits of space that are left actually in our membrane process. But thermodynamics says we want to get rid of internal space. We want to get rid of internal surface. So thermodynamics over time does everything it can to squeeze out all of the space that we're trying to put in. And so you have this process of very slow changes over time, which leads to a densification of the glassy structure. And that leads to reductions in permeability, often with increase in selectivity, though unfortunately not always. That's the sort of thing that engineers don't like. They don't like their materials to change over time. And yet these inevitably will to a greater or lesser extent. And so a lot of our research is about how we control that sort of thing, how we at least minimize it. And another thing which can occur is plasticization. If, for example, we're doing a gas separation, and one of the things we're looking at is how we can remove carbon dioxide, either from flue gases or from biogases or something like that. And carbon dioxide interacts very strongly with many polymers, including the ones that we look at. But where we have something like carbon dioxide which interacts with the stronger the polymer, it's also almost like a little a solvent for the polymers, where it changes the polymer. It creates more free volume, it creates more mobility just by having it there. And that in turn affects the permeation behavior. And so in practice, where plasticization occurs, what you tend to see is as the pressure that you apply to the feed increases, you start to get an increase in permeability and a loss of selectivity. And you also find that if you study a single gas on its own, which is what we often do in the laboratory because it's easy, everything is different when you look at a real mixture, and often worse. And so plasticization is another thing that we really have to think about, understand, and control. So, you know, when you're at university, you do things for publications, and there's lots of literature on membranes, gas separation membranes, evaporation membranes, where we're studying these sort of thick films, which might be 40 to 120 microns in thickness, in order to understand how a particular polymer or particular material behaves. But the problem we have is that when we try to go to a real membrane, we're now going to something which is very, very much thinner. In our case, it's often about 2 microns, but you can be aiming for less than a micron, even at just a few nanometers. And what we've measured in the thick film is not necessarily the same as what happens in the thin film, particularly dealing with something like a glassy polymer. So the properties of the polymer actually are different near a surface to in the bulk. As you go from a relatively thick film to a very thin film, the surface effects start to dominate. And particularly because of the sorts of things I'm going to be talking about, it's all about the conformational flexibility, how much freedom there is for the polymer. There's more near the surface, and so things are different in a thin film to the thick film. And so we actually need to learn a lot more about what happens when you get to these really thin films, which isn't always the same as what happens in the thick film. I mentioned physical aging as being one of the things we have to address. That is much, much faster in a thin film than in a thick film. So something which actually, in a thick film, it looks like it can easily stable over a couple of years, those changes can occur in a month or less in a thin film. Plasticization can be much more pronounced in a thin film than a thick film. So something which doesn't show much effect in a thick film can show a very pronounced effect in a thin film. And on top of that, we're trying to work with polymers which are as permeable as possible. So we're trying to get high permeability, which means we're talking about polymers which have a lot of extra volume, what we call free volume, high free volume polymers. And all of these things are much more significant in high free volume polymers. Since a lot more space and a lot more potential to lose it. So in terms of current research, we're very interested in what happens as we move from thick film to thin film and how we control some of these effects that occur when we're dealing with very thin films in a composite, for example. I mentioned physical aging, and there is a trick that is often played with glassy polymers, because it's just a physical effect. It's a densification occurring over time. It's his reverse. And so what we will often do is with many different polymers is often it's actually an alcohol, ethanol or methanol, just immerse it for a few hours or overnight, and that just swirls the polymer enough to effectively reverse many of the effects of aging. So it's like resetting it. Not necessarily quite the same as starting out, but it's hard to do that often with a thin-film composite because you can then get delamination of the different layers. But we can get the same effect actually just by storing in the methanol vapor. So a very simple way of keeping this in the methanol vapor can actually retard some of those aging effects or refreshen them. Which goes back to, again, what we do in the lab is often not representative of what happens in real life. So if you go to the literature and look at aging studies, what's normally happened, someone's in the lab, they do a permeation measurement on a membrane. Now, they're not going to leave that membrane in that bit of kit for months or years because they need the kit. So they take the membrane out, put it in a drawer, and weeks later, months later, years later, they go back, they measure it again. But in the meantime, that's been sat in the drawer. In real life, of course, a membrane is used with continuous permeation. And what happens under those circumstances may be very different to what happens when the membrane is sat in the drawer. And we need a lot more data, we've got very little data actually about what happens under ongoing conditions of use. So that's an area where we need a lot more information. But what we can say is that actually how you store things can make an enormous difference. And so one thing we've been playing with is storing in a methanol vapor atmosphere rather than just which has quite a reasonable effect. Okay, so that provides the context and the background and some of the issues. And now I'm going to focus on my favorite polymers, the polymers of intrinsic microporosity, or PIMs. And the story of PIMs, it goes back to the early 2000s when Neil McCoe and a friend who at that time was at Manchester University and I we were actually looking at trying to create networks which behave a bit like zeolites. And then along the way, we made something which was intended to be a model linear polymer to compare. And we realized we had a linear polymer which actually behaved in many respects like a zeolite or activating carbon. But it was processed. And that was really the start of the PIM story as it is today. And the first, well, we filed a patent application in 2003. The first publications came out in 2004 about this new class of robust solution processable nanoporous materials. And this term microporosity here, because people use that word in different ways, but we're using it in the sense that IUPAC defines in terms of absorption size, where the micropores are pores which are less than 2 nanometers in dimension. We can get really confused with engineers who think we're talking about pores of micron dimensions. So we have to be clear about what we're talking about. Patents go, I'm sure you know about these things in a place like service measurement systems. They're going to go through a long and involved process. The first commercial project I'll mention a bit about came out in 2014, but we're still exploring new applications. But the basic idea, what makes these polymers different to any other polymers? Now, the simplest idea of a polymer, which of course is a big molecule made up of lots of little molecules joined together. So in the simplest case, it's a long chain of repeat units. And most polymers, and of course, if we look around the room and look at closure rain, these are all polymers. Most polymers have backbones, which are a series of carbons joined by single bonds, or sometimes other atoms. And around those single bonds, you can get rotation. So we have conformation flexibility. That chain can wiggle around, and when it's in solution or when it's in the rubbery state, before you cool it down to form a glass, it can change its conformation. So a given chain can sample many, many different shapes over time. You can design polymers as a chemist which are very rigid, which can't change very much, but they tend to pack very effectively. If you end up with something very often, you can't process, can't do very much with it at all. Our trick is to try to make a polymer where you reduce the conformational flexibility. We have usually no, or something that's highly restricted rotational flexibility in the backbone. And we do that usually by building basically structures which are a series of rings, like a tiny little molecular ladder. But we also make the chain twist and turn, so it looks like a wiggly polymer, even though it can't wiggle. And we do that by including what we call cyclic contortion, something which puts a bend in the chain. And in the typical example that I'll show you, it's a spirocenter. So the point about this sort of structure is that we now have a polymer which can't pack down very easily or effectively. It can't crystallize. The shape that it has is fixed at polymerization, and although it can wiggle a little bit and flex a little bit and bend a bit, it can't undergo those large-scale conformational changes that a normal polymer can. And so it traps a lot more of what we call free volume, a lot more space in the solid state than a normal polymer. And the consequence of that is it behaves like a microporous material, where we're talking about material which behaves as though it has pores less than a couple of nanometers in dimensions. So the sort of the prime example, the prototypical example of PIM is the one that we call PIM1. It was the first one we made. We made a series of related polymers. And in the structure here, you can see that the backbone, it's all rings. It's either carbon rings or rings with a couple of oxygens in it. But it's all rings. There aren't any single bonds that rotationally occur around. But we have these units. That means that it goes to almost a 90-degree bend at intervals. And that comes from spirocenters. Spirocenters is where we have a tetrahedral carbon atom attached as part of two ring systems, in this case, 5-member ring systems. And so that sort of structure gives us this sort of wiggly, contorted structure, but without the conformational flexibility of a normal polymer. And in the case of PIM1, we started with 2 monomers, and this is one of the beauties, but that they're not cheap, particularly cheap, but they're commercially available. A lot of the other PIMs involve chemists spending an awful lot of time synthesizing bespoke monomers. But we have 2 monomers, one of which contains the spirocenter, and in this case, we're doing a bit of chemistry which is known as nucleophilic aromatic substitution. So we have a couple of hydroxyls on one molecule, a couple of fluorines or chlorines, nowadays we tend to work with chlorine as much as possible, on the other. And you'll see there's also a CN, a nitrile group there, which actually activates that monomer towards this type of chemistry. So it works very efficiently at temperatures that are lower than would normally be used for this type of chemistry. And so you can see on the screen the sort of the idealized structure of PIM1, which kind of assumes your polymer is a continuous sequence of the repeat units that you can see there. And in a moment, I'll talk about why actually in reality that may not be the case. But the thing about this is it's a polymer we can dissolve up in certain solvents. It's not always the nicest solvent, but we can dissolve them up in things like tetrahydrochlorine, chloroform, and such like, and therefore process it into powders. We can coat it onto surfaces. We can cast thin films just by letting the solvent evaporate. We can turn it into fibers by processes like electrospinning. And what you have in the end is it's a glassy polymer. We never see it in the rubbery state because we can get up to 350, 400 degrees C, and eventually, being a polymer, it can degrade. There are ways now of catching the glass transition just by having a very, very rapid jump. But we never see it in the rubbery state. It's always a glassy polymer in practice. But we can make it into coatings and films, and it has many of the properties of something like an activated carbon, but it's processable in a way that something like an activated carbon is not. And so one example of the way in which you might analyze a typical microporous material is by studying how nitrogen is absorbed at low temperature. So, and what you see here is at very low relative pressures. This is at 77 Kelvin, the temperature of liquid nitrogen. We get a very high uptake of nitrogen. And that's typical of a material with these very tiny pores that we're calling micropores. Because if you have, nitrogen, it doesn't interact very strongly with the surface. It's only relatively weak intermolecular forces. But if you put it in a little space where there's surface all around it, the interactions that there are get magnified. And that's why we're interested in these microporous materials. Even relatively weak interactions are magnified. You get much stronger sorption in the micropore than you would on a flat surface. And so that's very typical of things like zeolites and carbons, activated carbons. There are some differences as well. You'll see if you desorb the nitrogen, it's not the same as what happens when you adsorb. That's a process of hysteresis. And it's not the same sort of hysteresis. If you have material with slightly larger pores, what are called mesopores, 2 to 50 nanometers, then you get hysteresis, but it always closes at a relative pressure above 0.4. And where it closes depends on the size. This extends down to the low pressure region. That's very typical of these sorts of materials. And that's because this is a polymer which can actually swell when you cover penetrant, and also it's not a well-defined pore. This is free volume with a very complex sort of structure, so it's a complex micropore structure. Both of those things can give rise to this effect. But what we can see here is we take a typical sample of this PIM1 and prior type analysis, which I'm sure many of you know about, BT analysis, you can come up with a value of surface area, typically 700, 800 square meters per gram. That's not all real surface, but these are pores. This is the amount of nitrogen it would take up, which, if it were a flat surface, it would be that area. But it's indicative of how good these are at pulling in little molecules. And not just gas molecules. It loves little organic molecules. And so here we have an example. We have a dye in solution, it's a phenolic solution, and we're comparing actually 3 different high free volume polymers. But the one on the end, the PIM1, you'll see it's pulled all of the dye out of solution so that the yellow polymer has now turned green with the blue dye. So it loves many little organic molecules. And that actually is the reason for its first commercial application. Unfortunately, this is no longer available for reasons which have nothing to do with the polymer. But 3M created a little sensor which used PIM1, thin film of PIM1 at its heart. And this was used as an end of service life indicator for the cartridges they use for absorbing organic vapors that are used by people who work in environments with lots of organic vapors. And the idea is that this little sensor tells you how much of the carbon is used up and when it's time to change the cartridge. But we've been focusing on, in our research, very much on membrane applications. And I mentioned evaporation earlier, and this was actually the first membrane process that we looked at, simply because I've been looking at evaporation with zeolite membranes when this came along. Evaporation is a process, most polymers and most other materials, if you put them into a membrane, if you have an aqueous mixture, it's the water which transports preferentially. And that is used at present. It's used both with certain polymer membranes like polymer alcohol and also certain ceramic membranes. And you know, zeolite is really good at it. Hydrophilic evaporation is used at the moment in the processes for making absolute alcohol as part of the process, because it can be used to break in a zeotrope. What's much rarer is something which will pull an alcohol out of water. You know, after some waters you can mix them, separating them is much harder, and that's the basis of what we're trying to do. But this is one of the relatively few polymers which you can take a solution of alcohol, a dilute solution of alcohol, and then enrich it in the alcohol. In fact, we first looked at removing phenol from water. And later on, we looked quite a lot, for example, at butanol, with interest in biobutanol recovery after the fermentation process. So that's one of the ways in which we're looking at using these types of materials. But a lot of the research in the literature is about gas separations. And I said earlier that when we're thinking about membrane materials, we have this trade-off between selectivity and permeability. And back in 1991, a guy named Lloyd Robeson recognized this, and he found that it's quite a convenient way of comparing different materials. What he did is he took all the data that existed in 1991 for polymeric gas separation membranes, and he plotted the log of selectivity against the log of the permeability of the passes gas for a whole range of pairs of gases. And permeability, incidentally, I said before, that's normalized for the pressure difference and the thickness. And you'll see some rather odd units which don't relate to anything else in the world. The Bauer unit, which people use. People from Imperial, especially. No Bauer well. Yeah. But what Robeson is, he just drew a line through the best performing polymers. And that sort of showed the boundary of what was possible at that point in time. And the question then has been, can one improve on that? This is one example. This is air separator. This is the air separator. This is the oxygen-nitrogen gas pair. And so this is a current use for membranes. You probably don't realize how much safety might have depended on this, but one place where air separation membranes are used is on many aircraft. So if you've flown Boeing or Airbus, there's actually a membrane system on board. What it takes is it takes air and it throws away most of the oxygen to generate a nitrogen-rich inert gas. And that's then fed into the fuel tanks, so that as the fuel goes down, you don't get an explosive mixture above the fuel. And that's been done since, quite a few years ago, as a Pan Am flight exploded, and since then, or after that, it was introduced onto most commercial aircraft. So air separation is a current application. It's one where we'd actually very much like to get higher selectivity to open up a whole new range of applications, particularly applications where you want high oxygen content rather than throwing away the oxygen. But this sort of illustrates the point. So that red dashed line is where the situation was in 1991. And there are many points that Robeson looked at. I've just pointed out a polyamide, which is often used for these types of membranes, is macromide, and because it has a resource selectivity, but its permeability is pretty low. And there was a few polymers with very high permeability, in particular a substituted polyacetylene known as poly 1-trimethyl 1-propyne, PTMSP, very permeable, but not very selective. And after 1991, really there was not much change for quite a while. And then in 2005, when Leonard and also Karim Saad and Baligan were involved in the early days, when we first produced PIMs, we didn't have the facility to do gas separation measurements, and EFSA people were funding. So we collaborated with Detlef Fritsch at what was then known as GKSS. It's one of the Helmholtz centers in Germany, where they have a lot of experience in gas separation. And so in 2005, we published the first gas separation data and demonstrated the two PIMs, PIM1 and another one, PIM7, that we were much more permeable than most other polymers, but we broke that 99.1 upper bound. So, very little had changed for quite a long time. But then a few other polymers also started to appear, which were above that upper bound. So what Robeson did, not to be outdone, in 2008, he went back to the data and drew a new upper bound. So now, as chemists, we're now going to try and beat that. My friend Neil up in Edinburgh, and also people like Ingo Knau, who's now at KAUST, and a few other groups have done a lot of work on developing new PIMs. The same principle, the same sort of design ideas, but different ways of getting the rigidity in the backbone and different types of units to create the little kinks in the chains. And in particular, here are three examples of which are based around a unit known as tryptosine. And in 2015, this is actually Ingo Knau's group at KAUST, they decided they could draw new upper bounds, oxygen, nitrogen, and a number of other gas pairs, all based around new PIMs. In that paper, they didn't look at the carbon dioxide separations. That's always proved a little bit harder, in fact. But in 2019, we had an update there. Now, the carbon dioxide separations that people are particularly focused on, there's carbon dioxide and methane. That's because removal of carbon dioxide from natural gas is a current application of gas separation membranes, and also there's potential for upgrading biogas, which seems to have even more CO2 in it. And the other one of interest is the CO2, the carbon dioxide and nitrogen gas pair, because that's of interest for post-combustion carbon dioxide capture. And back in 1991, when Rosen really first looked at all of the data, nobody was thinking about carbon dioxide capture. So he didn't even look at the CO2 gas pair. By 2008, it was starting to become of interest because people started to become much more aware of global warming. And so there was a 2008 upper bound, which involved PIMs, of course, amongst other things, for CO2N2. And then in 2019, our friends up in Edinburgh, together with folk at ITMCNR, the Institute on Membrane Technology in Italy, produced a new upper bound, which is actually based, what they did is they had some new benzotrypsin-based PIMs, and they actually studied the aging behavior, and they used aged and data for different new PIMs to define new upper bounds for CO2 separations. So that both shows how things have changed over time. And remember, these are log-log plots, so there's some big changes in terms of what's potentially achievable here. And there's a lot of polymer science gone into this, but I like to think that PIMs have made, in the lab, made a big contribution to pushing the bounds of what is feasible. But we still have to address those things I talked about before. Can we control the aging? What is our plasticization? Can we actually make enough of some of these polymers, and really nice polymers, perform very well, but it's an awful many hours of PhD student work to get a few milligrams, and not necessarily easy to scale. We have to think about those issues of scalability of the materials. And that's one reason why, particularly in my group, we tended to focus on, we've gone back to the very first PIM one, and said, look, here's how you can make them two things which are at least commercially available. They'll never be super cheap. They could be made a lot cheap if you put the time into upscaling. What can we do with that by playing with the chemistry and playing with what to do? And one thing we've been interested in understanding much more is the role that topology may have played. Because I said before that if we look at the idealized structure of any of these polymers, it looks like it's just a linear sequence of repeat units. In reality, other things may go on. And what we have may be a complex mixture. And what we're showing is that for the same nominal polymer, just by varying the polymerization conditions a little bit, you can get very different results in terms of membrane performance. And so understanding how the polymerization, this is real chemistry, how the polymerization affects the polymer and affects what's going to happen in use, and optimizing that starts to become really important. So here's some things that we might actually have. You know, a polymer may not be a linear. It may very rarely actually be. The ends can join up, and you may have a side. In fact, of course, our polymers and many polymers are made by what's known as step growth polymerizations. And in principle, if you had a perfect step growth polymerization, you would end up with one enormous cycling. But other things may happen, Reese, I'll show you in a moment. If we get the branch, then we can start to create lots of other possibilities. We have tadpoles, different types of branch structure. We may go on to form networks, which may be open networks with ends that we can do things with, or closed networks. And all of these things can actually have different results in terms of how the polymer actually behaves, both in the short term and perhaps more significantly in the long term. How do we know what we've got? Not always easy. One tool which is very good at distinguishing different structures, but only up to a certain molecular weight. And of course, polymers are very high molecules, they're very big molecules. So we can't see the whole of this distribution that we have, but we can see up to a certain amount. And so we can actually play with the conditions, usually an excessive one component, to get a PIM1 which is more linear than most. We can see this here. This is matrix-assisted laser desorption ionization, time of flight, multi-top mass spectrometry. In this particular sample, we can tell that at least at the low molecular weight end of the distribution, we've got some actual linear ligands. Actually, in this case, we're using the chloromonomer, and with the chloro ends, we very rarely see the hydroxyl ends. They tend to they don't want to hang around. More commonly, actually, with any step polymerization, what you actually see in that molecular range is cyclics. And that's because if you're doing this sort of polymerization, if the end's joined up and you get a cyclic, nothing more is going to happen to it. If it's a linear ligand, it can go on to react. So if you push the reaction enough, what's left at low molecular weight tends to be the cyclics. So most step polymers, if you do multi-top, all you see is cyclics. It doesn't mean that it's cyclic over the whole distribution. A way of getting a bit more information is by looking at Markov plots. These are plots of the log of the intrinsic viscosity against the log of the molar mass. And you can get these days out of seismic 3D Poetry chromatography, where you have additional detectors. You have a light scattering detector, which gives you an absolute molecular weight without calibration, and you can have a viscosity detector, which tells you what your engine viscosity is. And in general, both with normal polymers and with PIMs, over quite a wide range, these plots of log intrinsic viscosity against log molar mass will usually be linear. The slope is telling you something about whether it's a random coil or a very stretched out thing, and normally whether it's a highly branched thing, though our branch PIMs behave differently to computers. But also where something is on that plot varies as you go from a linear to a more cyclic material. So we can have ways of getting whether we have a cyclic rich sample or not. And actually, if we have something which is linear PIM1, the BT surface area is actually much lower than the default. Because pretty much every polymerization model actually doesn't give you that. Other things are going on. Whereas cyclic PIM1 actually, highly rich PIM1 has a very high surface area. Because by tying the integrator, we've actually reduced the computational flexibility even further. But what about branches? In this polymerization, we're trying to be very clever and form two bonds effectively almost simultaneously. And usually this reaction is really, really efficient, linked with small molecules, that once you form one of the links which is going to make this cycle with two oxygens in, the other one goes quite efficiently. But that doesn't necessarily always happen. And particularly if your temperature is a little bit lower in the early stages of polymerization, when you're starting to build up polygon, you're more likely to get a monosubstituted structure rather than the disubstituted structure that we're looking for. And then further reaction then leads to a branch. Now we've got a branch structure. And we can actually see when that's happening if we look in detail very carefully at the proton NMR, because you get some little peaks to beside the main peaks for some of these carbons, some of these hydrogens, which we attribute to an unreacted or branched one. And we can do Lorentz peak fitting to come up with a sort of a rough degree of branching. And branch PIM1 behaves in some ways differently to less branch PIM1. For example, if we had palladium acetate, it forms a gel. Well, a more linear PIM1 won't. And in fact, if we analyze the bit of the polymer hasn't gelled, we see it hasn't got the branch points. So what's happening is we're getting actually coordination at those branch points. And paleomasticate is interesting because it can form triangles, it can form polymers. And so it looks like we're getting cross links, something like this. And we can use that. Now, the beauty of a PIM is also its downfall. The beauty of a PIM is that it's something that can process to form thin films and things, because it will dissolve. But something which will dissolve once will dissolve again. And so the very feature which makes it processable also is potentially a weak point if we want to use it with certain solvents. So I mentioned that we looked at evaporation, and we're very interested in organic mixtures, but there are many solvents in which a PIM on membrane isn't stable enough when you just cast it on membrane. But if we can cross-link it, we can stop it spreading too much. And the trouble is, there are many ways of cross-linking. We also lose free volume. But we have shown with this paleomastic method, we can create membranes which are stable enough to be used for evaporation of toluene dimethyl oxide, for example. And this is where there's a big volatility difference. In fact, we can pull pretty much almost pure toluene out of the mixture, and that's of interest in terms of the way in which the aromatic hydrocarbons are produced in trying to improve some of the processes. If we've got these branch points, we can actually get further reaction if you push it too far, and we can then start to build up different types of network structure. And again, that can have an influence on the properties. We can quantify the amount of network above a certain size just by simple filtration experiments. And at first we thought we wanted network because we can actually improve selectivity. But then, particularly when we're thinking about thin films, which I said things very different to thick films, sometimes we then get really bad aging behavior. So we think we've got an advantage in the short term. The student goes back a few months later and it's a disaster. But again, it all depends on how you do things. So here we've got a really bad example. This is exactly what you don't want in a thin film composite, where you've got a modest selectivity at the start and high permeance. Because this is a thin film composite, we're now looking in terms of permeance rather than permeability. And the odd unit for permeance is GPU, gas permeation, just as odd as that. But after just 28 days, we've not only lost a lot of our permeance, we've also lost selectivity. That's the last thing we want in the membrane. That's an absolute disaster. But we can play with the chemistry. We came up with a sample which we believe a sort of an average idea of the structure. Sorry, we've got lots of little cyclics and a few branch points. And these perform very, very much better. Over 120 days, we lost some permeability, but we've maintained selectivity and much, much slower processing. And the point here is that we can play with the polymerization. We can get different topologies. It can have a dramatic effect on what happens over time in the thin film of these polymers. And so learning how to optimize things is a key part of our current research. So, I think I'm nearly done. I'm getting carried away. Very, very quickly, other things that we're interested in, we can modify things chemically. For example, we can turn those nitriles into carboxylic acids. If we might come to take capture, in terms of selectivity and permeance, on the Robertson plot, there's an optimum range that you want to be in, depending on the process you're looking at. And this is an area that we've looked at. And just to illustrate some of the other things we have to think about, we can do an acid hydrolysis into a carboxylate-rich polymer, which seems to perform really, really well if we look at single gas emissions. But this is a thin film, and this is where plasticization can start to come. It doesn't look too bad on a thick film. It starts being really serious on a thin film, particularly interested in things that have hydrogen bonding. And that's a large part of what's going on. And so when you go to mixed gas, it's still actually quite good performance, but drops out of the range we're really looking for. And that's a plasticization effect. And so again, that's something that we're looking at, finding ways to improve on that. We can know what's possible, but we still have to play around to get the performance that we're looking at. Just go. Another thing we do is make mixtures to form what's known as mixed matrix membranes. There, it's all about getting compatibility between the different phases. And one way we've done that is by making a filler, which is effectively a variant of our own polymer. And also, we put bits of our polymer to improve the compatibility. And we can see that we can we can go from a situation where we have a big loss of permeability to a situation where we have a very tiny loss of permeability over 160 days, coupled with an increase in selectivity. So you can actually have quite a big effect on the results. There are a few other polymers, things which have been, many things have been used as additives. Just a few examples here. But just to finish, what I want to show is, if you just go back to the basic chemistry and produce a PIM1 with a better structure, you can have, at least in terms of selectivity, a bigger effect than any of the other things that you might have. You might add other things then to actually improve the permeance, but it pays to think about the chemistry of what you're doing. And so, to conclude, membrane processes give energy efficient electron separations, potentially at least. PIMs show promise. We're still looking at the best ways, but we have a number of good strategies for tackling the particular problems of physical agent and plasticization. Not all, even it may be the same label, but not all polymers are the same. Not every PIM1 is the same as every other PIM1. But there are a whole range of ways in which we can tailor performance. And so I want to say thanks for listening. Thanks to all members of my research group for the work they've put in over the years and a whole range of collaborations. But I'll mention a current collaboration which is important, which is the Sinhai Cell Program Grant, which links 6 universities across the UK, all involved in different aspects of membrane technology. So, any questions?
Yeah, I guess we will open it up to a brief Q&A. If you're listening online, please post them in the chat, the questions in the chat, and I'll try and get them asked to Peter. If I could.
We have the privilege of having you on site, so we'll try to give priority to questions from our listeners. But while those trickle in, and I'm sure we're getting those at the moment, is there any questions so far?
Yeah, so earlier when you were showing that nice straight line plots between selectivity and permeation, they were looking at quite idealized like binary mixtures. What would you take into like resistivity to something like the influence of humidity with some of these polymers, and how tertiary and other effects can have a huge impact on their performance?
They can have a huge impact on performance. Yeah. You know, often it's based on either single gas mixtures or simple binary mixtures. If it's talking about carbon dioxide capture, there will always be humidity present. There are not very many studies which have looked at that, just a few. So that's something we want to look at a lot more, and that's something where I think surface measurement systems are helping.
I've got a question online. The first one is, can you talk more about the 2014 first commercial PIMs on gas separation, sort of the application, material development, where they can find more information?
Okay, well, the commercial application was a sensor application rather than a separation application. And it was the sensor was actually devised by 3M, and they realized that PIM1 or a PIM was the material they needed, because what they wanted was something which would absorb like an activated carbon and be processed into thin film, which is exactly what we wanted. So they developed the sensor, but they licensed the PIM technology from us effectively. The way it works is there are a number of papers out there. Later I can send you references if necessary, but if you Google Neil Rakow, N-E-L-R-A-K-O-W, you should be able to track some down. It's quite a neat little sensor. What you have is, what you want to do is to know when vapors are getting past the activated carbon and into the sensor. And so you have a porous mirror next to the activated carbon, and then a very thin film of our polymer, and then a non-porous half mirror on the outside. And from the outside, light, some is reflected off the top surface, some goes through the polymer and is reflected off the bottom surface. That creates an interference color. And it's set up so that it looks green. If vapors, and it applies to a very wide range of organic vapors, things like styrene are particularly good, get past the carbon through the porous mirror into the PIM, that changes the refractive index, and that changes the color that you see.
Does it swirl the membrane?
It also swells it a tiny bit, which has the same effect, but it's mostly the refractive index change.
Thank you, Peter. We've got another question on, does the solvent for casting the polymer influence the porosity of PIMs? And if yes, how should the interaction between the solvent and the polymer be to increase porosity?
The solvent does have an effect. What's in the solvent has an effect. So if you're using chloroform, it makes a difference whether it's alcohol stabilized or aniline stabilized, because those little impurities are just the sorts of things that the PIM likes to take up. But it's not just about the solvent, it's also about how you're actually processing it. So when we make our thin film composites, we're using a coating process, and a lot depends on how fast the solvent evaporates. So it's not just the solvent, but also the solution concentration, many other things will affect what you get.
Good. And then there's a few more questions. So could you please elaborate on how a high network content can be prevented during synthesis of these polymers?
We've got a paper we've got a couple of papers on this, and probably get some more coming out. So you tend to get high network content when you get a lot of branching early on, and then you push the reaction so that you can get further reactions to form the four-way linkages that we showed. So it's a combination of the temperature profile in the early stages of polymerization and how far you push the reaction later on. So it's quite a delicate balance. And what we found, the way we found out some of this is, in the lab we use little heating blocks, and we found that if you just use different size heating blocks, you get different results, because that changes the temperature profile in the period when you're actually heating up to the nominal temperature of the polymerization. So it's quite complex. My Andrew Foster, who's here as a name on many of the papers, who's a postdoc with us, he spent a lot of time playing around with this. So he's the best person to contact for details of how to get what you want.
Okay, yeah. So contact Andrew Foster in Peter's group for the expertise in his group. Okay. And I guess you mainly focused on PIM1. In your opinion, can we expand your results to other types of PIMs, such as, I guess, troga-based polymer?
So, yes, the troga-based polymer is one of the other polymers that Neil has developed. So, some of the things are applicable. The chemistry is different, so you won't necessarily get the same sort of branching that we do when we're using the nucleophilic aromatic substitution. We haven't particularly looked at, we haven't studied that yet. But some of the things that I've talked about, not necessarily all of them, will be applicable.
Then one last one. So while PIMs can be used in different forms, Which form, in your opinion, is the best one from many perspectives for industrial application for large quantity gas separation, and why?
So in terms of membranes, we've been focused on thin film composite membranes. There are people, in fact Ryan Lively, who wrote that article I mentioned at the beginning, his group probably made more PIM1 than anyone else in the world, because he did better as a method for making hollow fibers. But it's not the easiest thing to do, and PIM1 isn't necessarily the best polymer, not least for cost reasons, for the underlying support. So we're focused on thin film composites. At the moment, working mainly with flat sheets, so in principle that can be applied to hollow fibers as well.
Good. Okay. Point, answer question.
Thanks for the presentation. Actually, when you got to the topology section, I'm a more chemist, well, chemical engineer turned chemist turned. I got a realization, almost I want to make a controversial statement, that maybe covalent organic frameworks are a subset of PIMs. I don't know how true that is. But what I really found interesting was the behavior with nitrogen at 77 Kelvin. It got me thinking, is the porosity the same at 77 Kelvin than it is at room temperature? Because I would imagine as you change temperature, particularly in a polymer, you might get changes in pore size, pore perhaps.
Almost certainly not. The problem is that we don't have the tools to really in fine detail analyze the porosity. So you can take something, you can do CO2 sorption at room temperature, and you can analyze the data and get pore size distributions, which actually do map onto what you get from nitrogen sorption to a certain extent. You get to lower pore sizes. So up to room temperature, it probably isn't very different. But when you go up to higher temperatures, particularly around 100, other things start to happen. And in fact, we've got some evidence from positron annihilation and other things that and there's also a dielectric transition which seems to occur in that region, that actually we go to a maximum effectively in terms of the porosity. So certainly there are interesting things going on at higher temperature. But by and large, within the level of what we're talking about, there's not much obvious change. The problem we have is, the permeation is incredibly sensitive to tiny, subtle differences in the pore size distribution. We do not have the tools to actually analyze those differences. So positron annihilation, you can actually see a tiny shift in an effective free volume element size, which is an order of magnitude change in permeability. And the permeability, particularly with PIMs, there's a difference between the permeability of the smallest gases, hydrogen and helium, which are probably through continuous free volume tracks, if you like, and the larger gases, which things are dominated by the bottlenecks in the structure. And that's actually what gives rise to a lot of the selectivity you get. It's that key difference. So you have to understand where those bottlenecks, what's going on. We don't have the tool to really analyze that yet. It's a lot we need to learn.
Thank you for interesting presentation here. I'm just understanding fundamentally the process of going on here when these molecules go into these structures. You're talking about pore sizes and pore spaces and the free volume. But I'm also wondering about the whole question of solubility here. Where do we separate when a molecule is going into a pore and when it's a solubility phenomenon? So I see a lot of what I see here is I'm thinking solubility, but I'm hearing porosity, and I'm wondering about that. And I'm also interested in the concept around the polymer topography and how critical that is. And it's a discussion to have here. I'm not sure the answer, but I'm curious what you're thinking.
That's a very, very good question. So the model which is usually applied to most of the membrane separation, so it applies to gas separations, osmosis as well. The model which is most commonly applied to separation in this region is the solution diffusion model. So permeability is the product of two things. There's a diffusion term and a solubility or sorption term. Now, the reason why we get a lot of our high permeability, particularly for CO2, is from the sorption term. So that's really, really important.
Yeah.
So for most polymers, the fastest gas is a hydrogen lead. So hydrogen is a tiny molecule. If it's just about diffusion, hydrogen is the fastest. In a PIM, CO2 is faster than hydrogen. That's because of the sorption component. So you cannot escape that. Now, it then gets even more complicated because when you're thinking about porosity, CO2 likes the really little, the ultramicron, the really little spaces. And so the pore size, well, we might call it pore size distribution of free volume elements. We're in a sort of magical world between a solid material and a porous material, where you can use the language.
Well, that's what I'm sort of wondering about, because I was particularly interested in the importance of the polymer topology, because we've done work modeling solubility of solvents in amorphous polymers, organic solvents, and it turns out the structural features of the amorphous polymer are uber critical to solubility.
Yeah.
I mean, so if you take polypropylene, and you look at the solubility of octane, polypropylene, it's amorphous, in amorphous only, it's very, very high. It's like 100%. Crystalline domain, but you've got these tight domains, which are constrained domains, and you get substantial solubility, but way less than fully amorphous, and way more than crystalline. So I suspect, or I'm wondering, that a lot of the performance enhancements or things you're seeing around changes in the polymer topography are core solubility of those molecules in those structures.
So when we're dealing with CO2, methane, hydrocarbons, the solubility tends to dominate what's in them. When we're dealing with oxygen, nitrogen, then generally they tend to be dominated by diffusion. So it is quite complex. And I've already said there's a difference between how the various formic acids behave. There's actually a change in the slope if you look at what happens with size. So I absolutely agree with you. The solubility term is really important, particularly because in some cases that's what dominates. When we're trying to improve, say, air separation, then probably the diffusion term is more.
But we use the molecules separate entirely.
So the solubility of sorption term is motivated.
Yes. Thank you.
Okay. Oh, can we have a round of applause for Peter? Yeah, I guess we'll end it here. Thanks again, Peter, for your wonderful talk. And join us for our next webinar when that comes out.
Very soon. Thank you again, everybody who attended online. We'll cut the feed here. Thanks again.